A very good morning to everyone. My name is T. Kim Boon. First of all, I would like to thank MPS and Malaysian Journal of Pharmacy for giving me the opportunity to present in this prestigious convention. The title of my study, Metabolomics of Metformin in Urine Samples of Healthy Volunteers. A little bit background about my study. Metformin is the first-line diabetic agent widely used for type 2 diabetes mellitus. It increases the glucose uptake in the liver and skeletal muscle and is suppressing gluconeogenesis in the liver and the interstitial glucose absorption. Lately, researchers found that the primary effect of metformin was reside in human gut. In the omics technology, it started with genomics, transcriptomics, proteomics, and now metabolomics. It is a study of substrate and products of metabolism that directly reflect the underlying biochemical activity and state of cells or tissue. In the targeted metabolomics, the metabolites are compared with non-reference standard, for example, pharmacokinetic study. In the non-targeted metabolomics, a comprehensive investigation of metabolites to identify pharmacological effects and adverse effects of a medicine. In the conventional phase 1 trial, it mainly targets pharmacokinetics performed by the pharmacologists or pharmacists and safety monitoring of single ascending dose and multiple ascending dose by the physicians. The dose escalation decisions in the phase 1 trial is highly depends on the combination input from the physicians, pharmacologists, and sponsor as written in the Malaysian phase 1 guidelines. In this study, we increase the value of pharmacologists by integrate metabolomics in phase 1 study, identifications of pharmacokinetics and dose-dependent pharmacodynamics effects through human metabolic pathway. The objective of my study is to identify the metabolic pathways of metformin in urine samples of healthy subjects. The methodology is divided into 3 parts: clinical, laboratory, and metabolomic statistical analysis. Healthy volunteers are screened for a list of inclusion and exclusion criteria. Eligible subjects are given metformin 1000 mg under fasting condition. We collect the pre-dose urine sample as U0, U1 is post-dose from 0 to 4 hours, U2 4 to 8 hours, and U3 8 to 12 hours. This is the short video about the clinical trial. This is our main research teams. And we conduct our trial at Clinical Investigation Center, UMMC. This is our research board. We perform clinical trial procedure briefing to the healthy volunteers and inform consent taken by principal investigator Dr. Rahman Ibrahim. Subjects screening activities, metformin tablets, dispensing, and also blood taking process. This is our urine samples, and we keep our urine sample in cryo valve before an analysis. Next is laboratory. We analyze the urine sample using the Agilent LC-MS Q-TOF with reverse phase column. The instrumental setting are using modified Matlin global metabolomics methods. We use pool quality control samples and internal standards, which are glycoside and atenolol, to ensure batch-to-batch consistency. The third part is metabolomics and statistical analysis. We use 3 software: GNPS, Metabolomics, and OmicsNet. For the chromatograms processing, we optimize the peaks using peak picking, peak alignment, peak gap filling, and peak annotation. Figure 1 is the total ion chromatograms for all the samples. We also perform retention time alignment using Orbi-Walk and Loess algorithm. Figure 2 is the retention time adjustment and difference plot. After this, we perform normalization based on probabilistic quotient normalization (PQN), log transformation, and range scaling. Figure 1 is the normalization of all the features to transform the features into normal or Gaussian curve for us to perform parametric analysis in the later part. We specifically extract the box plot for metformin, and we found that U0 sample does not contain metformin, but metformin are available in U1, U2, U3, and QC samples. Next is multivariate analysis. We are using principal component analysis (PCA) and heat map. Figure 1 is the heat map. The blue color is low intensity and the red color is high intensity. For the column, the green column is U0, the dark blue U1, light blue U2, and the pink is U3. The upper part for the U0 column, the features are mainly blue color, which is low intensity, and it changed to red color. At the lower part of the U0 features, they are in red color, which is high intensity, and change to blue color. Figure 2 are the PCA. We can see the QC are gathered at the center in red color. The green color is U0, slowly shift down to the dark blue color at U1, light blue U2, and the pink is U3. Next will be the univariate analysis. The data set are break down into 3 pair analysis, which are U0 versus U1, U0 versus U2, and U0 versus U3. We perform t-test with p-value of 0.05 and fold change analysis, and we combine both to become volcano plot. This is the figure 1 is the volcano plot, and the upper right hand sides are the up-regulated features, and the upper left hand sides are the down-regulated features. The tables are the number of features for positive and negative mood. After normalization, we have about 2,000 features in the positive mood and 2,200 features in the negative mood. In the pair analysis for U0 and U1, we have 800 features in the positive mood and 1,200 features in the negative mood, and so on. Result and discussion. We combine both positive and negative moods from the volcano plots to perform MS-PICS to pathway analysis. After 4 hours and 8 hours post-dose, urea cycle or amino group metabolism, glycine, serine, alanine, and threonine metabolism are having significant biological perturbation under the control environment after metformin administration. Similar metabolic pathway were observed by other studies. Figure 1 shown pathway analysis for metformin using enrichment factor. Figure 2 is the 3D visualization for both of the metabolic pathway. We further extract the positive mood for the enrichment analysis. We find that 5 pathways are significantly found. Galactose in blue color, tryptophans in red color, tyrosine metabolism green, arachidonic acid in purple, and arginine and proline in pink color. This is the 3D pathway. We also perform features in negative mood for the enrichment analysis. 5 pathways are significantly found, which are pentose and glucuronate interconversion, purine, phenylalanine, tyrosine, and pentose phosphate pathway. Figure 2 is the same result in the network analysis graph. Last, conclusion. Metformin was absorbed and excreted through urine, as shown in metformin box plot. Untargeted metabolomics able to identify human metabolic pathway of metformin according to time and dose. Global metabolomics is a holistic approach to identify various pharmacological effects of the drug during phase 1 clinical trials. As you all know, Malaysian guidelines for phase 1 unit inspection has been established in 2018. In fact, last week, our DG has officially launched National Research Volunteer Register, which mainly target phase 1 trial. We will expect influx of phase 1 trial in the next few years. We wish that more pharmacists could step up to become a phase 1 clinical trial pharmacist and involved in dose escalation decisions. Acknowledgement. I would like to acknowledge University of Malaya and Ministry of Health of Malaysia. I would like to thank my research team, Associate Prof. Dr. Hasniza, Dr. Luqman, Dr. Najiha, Dr. Muhammad Zuwari, Dr. Zaria, Kashfu, Shaza, Azwa, and many other people who make the research success. This is my references, and the abstracts for the plasma result has been published in the Journal of Metabolism, Clinical and Experimental. I would like to welcome you to visit the poster 0047. Thank you.